Hello, Eastview Scholars. On this edition of Sharp Shorts, we're going to look at the cell cycle. And then in subsequent short little tutorial films right after this, we'll also talk about mitosis and then controlling the cell cycle and how it controls of and how it can lead to cancer. Anyhow, what is the cell cycle? Well, it's just the lifespan of the cycle of the cell. And whether you're unicellular or multicellular, whether as a unicellular organism you need to reproduce yourself or clone yourself essentially, or as a multicellular organism you need to develop into a full multicellular organism from a single fertilized cell. You need to grow both until maturity, and then growth also includes replacing old cells, etc. And then you have to repair if you get damaged. And that whole process is called the cell cycle. So from when I start really until when I as a cell divide again and have the parent cell and daughter cell or become two cells, that's a cell cycle. Okay. And you can see here examples of here's reproduction, here's growth and development, here's a little tissue renewal even closer. You can see here from a single fertilized cell that you become a multicellular organism here. Okay, and that requires a cell cycle. The things about the cell cycle that we have to keep understanding is a couple things. The big function of it is every cell, regardless of singular, unicellular, or eukaryotic, is that each cell must have the full complement of DNA. And so the process of dividing the cell has to preserve the integrity of the DNA and make sure that that's appropriately divided completely. And there you have to copy the DNA then and go that way. So it's just making sure through checks and balances that you have the whole genome in everyone, and that's packaged in chromosomes. Okay. The only time that, and we should say here, the only time that you don't get the full complement of DNA is in meiosis, which produces the sex cells. Okay, so what you can see here is here's a normal somatic cell. This would be the fertilized cell that went on, and that's a normal cell, and that's the diploid number. So for you as a human, 23 from mom, 23 from dad, 46, that's your normal complement. That then, as you develop into an organism and as those cells replace themselves, skin cells, etc., replace themselves, they need a method to make sure that you maintain in each cell those 46 chromosomes. Now in meiosis, which we'll have another film on, which is a different type of cell division, the key to that is you have to be haploid so that when you do fertilize, that you don't give too much or too little DNA to the fertilized cell. Okay, but here again, here's chromosomes condensing. So the chromosomes need to condense, and then they need to have a method during the division to divide equally. Okay, and that's really the key to the cycle. One thing to understand about the DNA is it does condense. You can't normally see it under the microscope until it gets into what's called the mitotic phase, the dividing phase. When you do make a copy of it, then those two copies stay attached to each other. And then the key to that will be them separating during a certain phase of mitosis so that each cell can get and preserve the integrity of it. As they're attached by what is called the centromere, each one is called the sister chromatid, and then they will divide and become chromosomes, as you can see here. So here is the copied DNA with that typical kind of X thing that they show. Okay, here's the centromere. There's each one's a sister chromatid, and then the process of separating will make sure that they all stay together and keep their integrity. During the cell division itself, that's the key. You need to copy the DNA before the mitotic phase, and then have a method for those sister chromatids to separate so that each nucleus gets the full complement of DNA. And then the cytokinesis portion of the cell division is dividing the cytoplasm and the organelles that are within it. Gametes, we said, are divided by, go through meiosis. which does not yield identical cells. And we'll talk about that when we talk about meiosis. Okay, so here are the phases. The two phases are interphase and the mitotic phase. And we've given you an idea of the mitotic phase. It's important is preserving the integrity and making sure each cell has the full complement of DNA. Interphase is really the growth phase, and then when needed in preparing for mitosis, that you copy the DNA. A couple things to understand. The cell cycle depends on the cell. How long it takes, when it's triggered, what it needs to do depends on the cell. Neural cells don't. They're amitotic. They don't divide. So once a neural cell is fully grown, it goes into what's called a G0 phase, which we'll talk about later. But that basically puts it into, I don't want to use the word dormant, but it's just a non-copying phase. Dormant makes it sound like it's not doing anything. Even when cells aren't going through mitosis, they're doing normal cell functions. Even as they're growing and doing their stuff, they are doing what the cell needs to do. It's just waiting for those cell controls and triggers to tell them to divide. And that might be, it's very often, much more often as a developing embryo, and then as you, obviously it's very busy as a child and a teen, but then as you get to be an adult, that cell cycle may change depending on the type of cell and the growth and your age. Okay, so interphase is really most of it. The growth phase, copying DNA when that's triggered. The second, gap phase or growth phase, preparing it for mitosis. All good growth phases. The initial one, the G1 phase, the first gap phase, obviously when you've gone through mitosis, you have two smaller, not full-size cells, and they need to grow back, and they need to prepare themselves for doing cell functions and stuff like that. But then there's a trigger, which we'll talk under controls as to when it goes into the S phase, because once it passes the G1 phase, it is going to divide. So this is kind of the controlling phase right there. Okay, so here's the illustration. There is your G1 phase. That's, you can see it happens right after mitosis has occurred. There is your DNA copying phase. It's still growing, but that's when the DNA is copied. And there's your final growth and gap phase. And again, once we pass this point, this is all going to go through. And that's the interphase portion, which is 90% of the cell cycle. The other, the mitotic phase, which we talked about, the division, is split into one or two depending on the scientist you want to talk to. The actual dividing phase and the cytokinesis, which is dividing the rest of the cytoplasm and the organelles. But that produces two cells, one a parent cell and one the daughter cell. And there we go. And there's our phases of mitosis. What we don't have in here is pro-metaphase, which is before, in between prophase and metaphase. I don't have that typed in here. When you're looking under the microscope, it's very hard to tell. But if you understand the thing, this is like early on when the chromosomes condense. And as we'll see with phases of mitosis in our next film, pro-metaphase is when the nuclear envelope is dissolved. Anyhow, that is our point. In our next film, we're going to look here, as you can see, this nice colored micrograph of what is the process here of mitosis and how does it work. But anyhow, that allows us to understand the cell cycle. Scholar up, Eastview.